Hello everyone, welcome to my YouTube channel Simple and Easy Zoology. In this video tutorial, I am going to talk about one of the main function of the plasma membrane, that is membrane transport. You know that the plasma membrane has a capacity to transport many material across it like water, gases, mineral, nutrients, ions, amino acids, carbohydrate, etc. Anyhow, the transport of all this material across the membrane is not that easy, isn't it? As I discussed in the last video, some of the substances like small and non-polar, they can easily pass through the membrane. On the other hand, the larger and polar molecule face difficulty passing the membrane, isn't it? So, to solve this problem, the plasma membrane has developed different methods of transport for different types of substances. So, mainly there are two types of transport, passive transport and active transport. So, in this video, I am going to talk about passive transport. In passive transport, it is a movement of molecule from a region of high concentration to a region of low concentration across a semipermeable membrane without expenditure of any energy. So that is the definition of passive transport. We'll know the meaning of it by taking an example. So if you imagine this as a cell and this is a plasma membrane which is present around the cell. So say for about 20 molecules of oxygen is present outside the cell and of about 2 molecules of oxygen is present inside the cell. According to the definition of passive transport, it is a movement of molecule from a region of high concentration. Here the oxygen molecule is present more outside when it is compared to inside, isn't it? So the oxygen molecule will move from a region of its high concentration to a region of its low concentration, that is from outside to inside. So along the concentration gradient or down the concentration gradient, this oxygen molecule will move. So that is according to the definition of passive transport. So the main feature of passive transport is the movement of molecule always takes place from high concentration region to low concentration region. There is no expenditure of energy. The movement of molecule may takes place with or without the membrane, and the movement of molecule will be slow when it is compared to active transport. And the last one is the movement of molecule across the membrane takes place until it reaches the equilibrium. So what do you mean by equilibrium? If you consider this example, there are 20 molecules of oxygen is present outside and 2 molecules of oxygen inside, isn't it? And you know that the movement takes place down the concentration gradient, that is from outside to inside. So till what the movement takes place? Till it reaches the equilibrium, means the number of molecules which is present outside the cell and inside the cell should be equal. So once it reaches that condition, the movement will stop. Say for about 9 molecules of oxygen has entered from outside to inside. So how many number of oxygen is present inside now? That is 9 plus 2, 11. And outside 20 minus 9, that is 11. So both inside and outside 11 molecule of oxygen is present. It means it attains equilibrium. So once it attains equilibrium, there is no net movement of molecule. Okay, so these are the features of passive transport. So there are mainly 3 types of passive transport: osmosis, simple diffusion and facilitated diffusion. So now I am going to talk about the concept of osmosis. The word osmosis is derived from the Greek word osmos. Osmos means pushing. Okay, in osmosis, it is a movement of solvent molecule. What do you mean by solvent? It is the one which dissolves solute in it, isn't it? And the best example for the solvent is water. So water is a universal solvent. So in osmosis, it is the movement of solvent or water molecule from a region of its high concentration. Again, the word its is very important here. Its means it is a concentration of solvent or concentration of water. So from its higher concentration region to its lower concentration region across a permeable membrane is called as osmosis. Osmosis can be easily demonstrated by this experiment using thistle funnel. Thistle funnel having broad mouth and narrow stem is taken and its broad mouth is tied with the egg membrane using thread. Here the egg membrane acts as a semipermeable membrane. So what do you mean by semipermeable membrane? It is the one which allows only smaller molecules to pass through it but not larger. Or in other words, it is the one which allows only solvent molecules to pass through it but not solute. So after tying the egg membrane, it is filled with the 10% of sugar solution. And the initial level of the sugar solution is marked. Okay. Later this setup is placed in a beaker containing water. And the water initial level is also marked. So in the beaker, say for about 100 molecules of water is present. So no solute is present in this water because it is a pure water, isn't it? And again say of about 100 molecules of water is present in this sugar solution. So out of 100, around 10 molecules of sugar is present, isn't it? So 10 molecules of sugar is associated with the 10 molecules of water. So how many free water molecule is present here? 90 free molecule is present in the thistle funnel containing sugar solution, whereas 100 free molecule is present in the beaker. Okay. Later this setup is clamped to a stand and it is kept undisturbed for few hours. So after few hours, when you observe the setup, there is an increase in the sugar solution level in the thistle funnel and a decrease in the water level in the beaker. From this one can conclude that the water molecule which is present in the beaker has moved into the thistle funnel by crossing the egg membrane. So it is nothing but the osmosis, isn't it? So movement of molecule from a region of its high concentration. So high concentration of water is present in beaker to a region of its low concentration across a semipermeable membrane is nothing but the osmosis. From this experiment, osmosis can also be defined as it is a movement of water molecule from a region of less solute concentration. So how many solute is present in this? 0. So from a region of less solute concentration to a region of high solute concentration. How many solute is present in this? 10. Okay. So it is a movement of water molecule or the solvent molecule from a region of less solute concentration to a region of high solute concentration across a semipermeable membrane is called as osmosis. So these are the definition of osmosis. You can see the phenomenon of osmosis in your day-to-day life, and the best example for that is swelling of raisin. So what happens when you soak a raisin in a beaker containing water? After a few hours, the raisin will bulge, isn't it? Why? That is because of osmosis. So water from a region of its low solute concentration, that is from outside, it entered into the high solute concentration, that is inside the cells of raisin. Okay, so osmosis has taken place. It results in bulging of the raisin, and this type of osmosis is called as endosmosis. It is a type of osmosis where the movement of water molecule takes place into the cell. As a result of endosmosis, what happens? There will be increase in the concentration of water molecule inside the cell, and this molecule exerts a pressure on the walls or plasma membrane of the cell, and this pressure exerted by the water is called as hydrostatic pressure. Okay, hydrostatic pressure, because it is exerted by the water molecule, and the hydrostatic pressure which is caused due to osmosis, it is called as osmotic pressure. Here hydrostatic pressure is exerted due to endosmosis, isn't it? That pressure is called as osmotic pressure. So osmotic pressure can also be defined as it is a minimum pressure that we apply to a solution to avoid the entry of solvent into the solution across the membrane. Okay, it is a one more definition of osmotic pressure. And this osmotic pressure play a very important role in the absorption of water in human being, like the absorption of water in the large intestine of human, or reabsorption of water in the kidney, or uptake of water from the capillary. These all phenomenon takes place due to osmotic pressure. So that is regarding endosmosis. One more type of osmosis is exosmosis. And the best example for that is pickle. What we will do while preparing the pickle, we will soak a tender mango in a salt or salt solution. So as a result of that, the mango will shrink. Why it will shrink? Because of exosmosis. Then what do you mean by exosmosis? It is a movement of molecule out of the cell. In this example, the water molecule from a region of its less solute concentration, that is from inside the tender mango, has entered into a region of high solute concentration, that is outside. So it results in exosmosis, due to which the tender mango shrinks. Okay, that is the best example for exosmosis. One more important concept in the osmosis is tonicity. Tonicity means it is a strength of the solution or solute concentration of the solution. Based on that, there are three types of a solution. First one is isotonic solution, hypotonic solution and hypertonic solution. We will discuss all these three types one by one. First one, isotonic solution. As a prefix, iso refers to the same. So, this type of solution have same type of solute concentration when it is compared to solute concentration of a cell. So, what happens when you place a cell in isotonic solution? There is no net movement of water molecule, isn't it? Because solute concentration outside the cell and inside the cell is same. So, there is no net movement of a water molecule. Okay. And the best example for that is 0.9% of NaCl. So this 0.9% of NaCl acts as isotonic solution for RBC. Okay, when you add a drop of blood to a 0.9% of NaCl and when you observe this RBC under microscope, there is no change in the shape or size of the RBC molecule because there is no net movement of water molecule in and out of the RBC cells. Okay, that is regarding isotonic solution. Second type is hypotonic solution. So as a prefix, hypo means less. So in this kind of solution have less solute concentration when it is compared to solute concentration of a cell. So what happens when you place a cell in the hypotonic solution? Endosmosis takes place, isn't it? Because the water molecule from a region of less solute concentration, that is from hypotonic solution, it enters to a region of high solute concentration, that is inside the cell. So that is nothing but the endosmosis. So due to endosmosis, the pressure inside the cell increases. So since the animal cell don't have cell wall, when there is increase in the pressure inside the cell may burst. On the other hand, the plant cell have cell wall. So when the plant cell is immersed in the hypertonic solution, what happens? Endosmosis takes place. But the plant cell won't burst because it have rigid cell wall. So in the plant cell, due to endosmosis, the pressure increases. And that pressure is called as Turgor pressure. It is nothing but the hydrostatic pressure. But in the plant, it is called as Turgor pressure. And the Turgor pressure is very important in keeping the plant erect position. You might have observed when you didn't water a plant for a day. So what happens? The plant will shrink or it will bend, isn't it? Why? Because of decrease in the Turgor pressure. Again, when you water it, what happens? The plant will come to normal position because the water enter into each cell and there will be increase in the Turgor pressure of each cell. So because of which the plant comes to normal position. And the best example for hypotonic solution is solution having concentration less than 0.9% of NaCl. So this solution acts as a hypotonic solution for RBC. So when you add a drop of blood to hypotonic solution, what happens? Endosmosis takes place. The cell will bulge and ultimately it will burst because it doesn't have cell wall. Okay, that is regarding hypotonic solution. Third one, hypertonic solution. As a prefix, hyper refers to more. So this kind of solution have more solute concentration when it is compared to solute concentration of a cell. So what happens when you place a cell in a hypertonic solution? exosmosis takes place. That is, water from a low solute concentration, that is from inside the cell, it enters into high solute concentration, that is to the hypertonic solution. So exosmosis takes place, which results in shrinkage of the cell. Okay. And the best example for this is solution having the concentration more than 0.9% of NaCl. So this solution acts as a hypertonic solution for RBC. So when you place a drop of RBC to the hypertonic solution, what happens? The RBC will shrink, and that condition is called as crenation. Crenation means shrinkage of RBC. Okay. What happens when you place a plant cell in hypertonic solution? Again, exosmosis takes place. So plant cell will have large central vacuole which consists of more amount of water. So water from the central vacuole comes out, because of which the plasma membrane separate from the cell wall and it will shrink. That condition is called as plasmolysis. So plasmolysis is separation of plasma membrane from the cell wall due to exosmosis. Okay. So that is all about the osmosis. In the next video, I am going to talk about the simple diffusion and the facilitated diffusion. I hope you like this video. If you like this video, please hit the like button and subscribe to my channel. Thank you.